Okay, so today we are going to start our process with going over the cell membrane transport. I just drew a small part of our cell membrane here. This is not our whole cell membrane. So what I did here, guys, if this is our cell membrane of our cell, and you remember we went over the cell membrane in a previous video, what I did was I just took a very small portion of it and I magnified it so we can, see just a part of it better. But regardless of this, guys, our cell membrane is going to be made out of this all over. It's not two-dimensional where it's just surrounding here. It's going to be in front. It's going to be like a whole spherical object. So this would cover the whole sphere of our cell membrane. I'm going to go over the individual parts with you and we'll start there. Now, when we were talking about the macromolecules in the previous video, we talked about the polymers of lipids, and there was 4 of them. We talked about waxes, steroids, triglycerides, which we went over in that video, and lastly, we talked about phospholipids, and I said we'll talk about phospholipids later, and here it is. So, these things here that are black and red, these are our phospholipids. So let me explain to you what their composition is. So we have 2 parts to our phospholipid. We have a circle, and then attached to that, we have 2 tails. Okay? Now, the circular part of our phospholipid, this is called the phosphate. And the phosphate is hydrophilic. Okay? What that means, with it being hydrophilic, it likes water. It can get real close to water. Water will form hydrogen bonds with the phosphate group. In other words, it can get real nice and close to it. Okay? Let me erase my cell membrane here. Our second part is going to be this bottom area here. These are going to be, and there's 2 of them, fatty acid tails. And if you remember, our fatty acids, these were the monomers of lipids, okay? So 2 fatty acid tails, and these are hydrophobic. Hydrophobic. So they don't like water. That's the phobia of water, okay? Hydro means water, phobic, fear of, all right? Literally means, water fearing. So if we were to put water on one part of this cell membrane, let me explain to you what would happen here. So first off, let's say this area up here is going to be outside of the cell. Down here, we're going to say this is inside of the cell. And you can see that there are 2 layers of phospholipids making this up, and the phosphates are pointing to outside the cell, and the phosphates are pointing to inside the cell. And then we have a double layer of fatty acids in the middle. But if we put water in here, the water would get up close and personal to our phosphate groups, okay, the phosphate heads there. It would get right up against that. But the second it tried to squeeze through, it would get repelled by the fatty acid tails, and the water would have to turn backward. So, moral of this story, guys, is that water cannot pass through the phospholipid portion of the cell membrane. Remember, water is polar, and the fatty acid tails, they're nonpolar. So they are not going to be combined. Nonpolar and polar actually separate. That's why water and oil separate. Okay? All right. So now let's get into our cell membrane transport. Okay? When we are doing cell membrane transport, in other words, bringing things into the cell or things leaving the cell, there's 2 terms we have to talk about. The first one is going to be passive transport. Now, what we mean by passive transport, it does not require energy to occur. So in other words, no ATP is needed for this, all right? Now, passive transport, there's 3 different types we're going to go over, and in this video, we're only going to go over 2. The first one is just diffusion, or you can call it simple diffusion. The second one is called facilitated diffusion. And these are the 2 we're going to go over in this video. And our last one, which will be our next video, is going to be osmosis. Okay, so for passive transport, it does not require energy. It's going to go with the concentration gradient. In other words, all of these things are going to go from high to low concentration. And we'll talk about that a little bit more, and I'll diagram that out for you. Our second type of transport is called active transport. And this one requires ATP. All right, so active transport, we need ATP for that. And I don't even have to number any of them because, yeah, we are only going to have one type of active transport. However, the one thing I want to mention with active transport, the main one you're going to have to know is going to be Na+, which is sodium, and K+, which is potassium. We'll go over the sodium-potassium pump, okay? All right, so like I said, this video, simple diffusion, facilitated diffusion, and active transport, and that's it, all right? So let's start with our simple diffusion, and I'll draw that over here. And we'll make a little line going down here. Okay, so simple diffusion is only going to be on this side. All right, we'll do something else over here. So for simple diffusion, here's the things we got to know. This is again going to move from high to low because we don't need energy for it. And we're going to be moving small, non-polar molecules. All right, things like oxygen and carbon dioxide. All right. Okay, so let me draw this out for you guys. So let's say on the inside of our cell, we have a few oxygens. O2, O2. Okay. Outside of the cell, we just took a nice deep breath in. We have a whole lot of oxygens here. I'm going to erase this outside of the cell just so I have a little bit more room, but it still stands true that is outside of the cell. All right. Make one more. Okay, so looking here, guys, I have oxygen. Just know oxygen, carbon dioxide, these are small nonpolar molecules. When they're small and nonpolar, what that means is they can pass right through these phospholipids, okay? So they go through the phospholipids. Okay. Now, count them on each side. 1, 2, 3 inside the cell. 1, 2, 3, 4, 5, 6, 7. So I have 10 molecules of oxygen altogether. We want the same amount on each side. So if you do 10 divided by 2, you get 5. I have 3 on this side. I have 7 on this side. I need 5 to be on each side. So what we're going to do, let me get a different color here. We're going to take 2 of these ones right here. And we are going to go right through the phospholipids and bring them over to this side, okay? And then when we do that, there's our 2. They're no longer here. So now I have 1, 2, 3, 4, 5 on that side, 1, 2, 3, 4, 5 on that side. Once we have 5 on each side, we have reached something called equilibrium. That is when we have the same amount of oxygens on each side of our cell membrane, okay? And that's our goal for all of passive transport. Our goal is to have the same amount of something or the same concentration of something on each side of our cell membrane, okay? So that's simple diffusion. Small nonpolar molecules moves through the phospholipids. Always going to go from high concentration, because we had 7 up here, we had more up here, that's high concentration, to low. So we had 3 down there, all right? Okay, next one. This is going to be facilitated diffusion. And we'll make a little line here to give us some room. So we'll concentrate on this for our second one. This is again going to still move high to low. Difference, we are going to be moving large polar molecules, or we're also going to be moving ions. So what is an ion? Well, here's what an ion is. You can see we have oxygen over here. It's just an element, two oxygens bonded together. We have carbon dioxide, but there's no charges on them. Let me show you what an atom looks like if it has a charge to it. Let me draw chlorine. Chlorine has a negative charge. I got that negative symbol up there. That tells me it's an ion. Another one, magnesium. Mg with a 2 positive charge. That's an ion, okay? So anytime you see something with a charge, it's an ion, all right? Other things, large polar molecules, glucose, fructose, galactose, okay? All those are large polar molecules. Now, because they are polar, just like we talked about water earlier, water cannot pass through the cell membrane because it gets repelled by these fatty acid tails in the middle. So here's what we're going to do. Let me draw some glucose here. Glucose, it's polar. It can't get through the phospholipids here. So I'm going to draw some glucose. I'm not going to label them all, but these little brown, larger circles are glucose. All right, so 1, 2, 3, 4, 5, 6, 7, 8. And we'll draw one more. There's 9. And I'm going to draw one outside of the cell. Okay. Now, because these glucose molecules are very large, they can't fit through our phospholipids. This is where this guy's going to come into play. This is a membrane protein. And what that membrane protein is going to do is allow these larger polar molecules to pass through. So I got, like I said, 9 on this side. This is going to be our high concentration. I got 1 on this side. That's going to be our low concentration. 9 plus 1 is 10. 10 divided by 2, still 5. So I need to get 5 on both sides. So what I'm going to do is I'm going to circle all these ones, and we're going to bring them through this membrane protein. And now they will go up there. Okay, so again, they move through a membrane channel or a membrane protein. Cool. All right, our last one we're going to go over here is going to be active transport. So here's the difference between all three of them. Simple diffusion, we're going to move right through the phospholipids. It's going to go high to low, and we're going to move small nonpolar molecules. Facilitated diffusion, we're still going high to low. Nothing changed there. The only difference is instead of going through the phospholipids, we got to go through a membrane protein because these molecules are larger or polar. Active transport, completely different scenario. So we are still going to be moving large polar molecules or ions, but we are going to be going from low to high concentration. And we're also going to be moving through a carrier protein, and I'll show you what that is here in a minute. And lastly, we are going to need ATP for that. All right. So let me give you the classic example here, guys. This is going to be the sodium potassium pump here. Now, what I'm going to do is I'm going to start off, and we are going to have a little bit of sodium. Whoops. We're going to have a little bit of sodium inside of our cell. Okay, so the little triangle guys are sodium. I'm not going to label them all up here. I'm just going to make them into little triangles for us. And we're going to have a good bit of sodium outside of the cell. So you can see this is low concentration, this is high concentration for our sodium. Now let's just talk about sodium for a minute. Do we want sodium inside of our cells? And that answer is absolutely not, because if we have a lot of sodium, a lot of salt inside of our cell, wherever salt goes, or wherever sodium goes, water follows. So if we have a whole lot of salt in our cell, the water will follow it. And if water follows sodium, our cell's going to get gigantic, huge. and it can go through a process called cytolysis, where the cell actually gets so big, so filled with water, it bursts and that cell dies. And we don't want that, okay? Let me draw some potassium for us here. So here is our potassium. I'm going to make it into circles. And we're going to have a good bit of potassium inside of our cells and just a little bit outside. Cool. All right. Potassium does not make water follow it. So what we want to do, guys, is we are going to get this Na and we are going to bind it into these three little areas on our carrier protein here. So this is Na, this is Na, and this is Na. And what we're going to do is we are going to expel that outside of the cell. It was these three sodiums right here that latched on into that area and got expelled out, okay? Now, this thing, I can't animate it, show you it in real time, but here's what happens to it. It opens up on one side and collects whatever, sodium, potassium, whatever it needs, closes, spits it out on the other side when it opens on the other side, takes in whatever it needs on that side, closes, opens, like that, okay? So it keeps functioning like that. Where the channel protein over here, it's always kind of open, all right? Okay, next, we're going to take some of these potassiums. We're going to lock them into this side, right here. And once it opens on this side, these potassiums will be spit into the cell. Okay, so that is active transport. We are going from low to high concentration. We want sodium outside of the cell. We want to bring potassium into the cell to try to keep those charges the same, because they both have a positive charge. All right. Now, the last thing I got to draw with this is, in order for this to happen, we need to bring in, for each time we do this, we need to bring in some whoops. There we go. We need to bring in some ATP. We need to bring in some energy. If we do not have energy, we cannot go from low to high concentration. All right, we can only go from high to low, just like we did with facilitated diffusion and simple diffusion. Okay, so end of this, guys, we move 3 sodiums out for every 2 potassiums we move into the cell. And again, ATP is required. All right, so in this video, guys, we went over simple diffusion, facilitated diffusion, and active transport. Next video is just going to be on osmosis. It's a little bit of a different process than this one, so we'll save that for another video. Have a good rest of your day, everyone.